In this advanced higher video, what we're going to be looking at is the ways, different types of data, the ways that data can be represented, and what you can do to interpret that data. So firstly, we need to know the different types of variable and data that we can get. First thing we have are discrete data or variables. These are variables that can only have fixed values and you fit into a particular group. So a good example is your shoe size or sex. Continuous data, on the other hand, is data where you exist on a spectrum. Height and weight are good examples of these. We can also have qualitative data, and this is data that cannot be measured in numbers at all. Then we have quantitative data, and because it has quantity essentially as part of the word, that is data that is and can be recorded as numerical values. As a subset of quantitative data, what we can do is take quantitative data and turn it into ranked data. This is where we take all of our numerical values and organize them from smallest to largest, and then based on the size of the number and its position, we give it a rank. So our example here is if we did 3 experiments, one value gave us 2.2, one gave us 5.9, and one gave us 0.3. If we were to organize those from smallest to largest, we would give 0.3 the rank of 1, 2.2 the rank of 2, and 5.9 the rank of 3. Next thing we're going to look at are scatter graphs and plots, which are a really common representation of data that you would get in one of the supplementary sheet-based questions where you have to answer questions based solely on data provided to you during the exam. Now, scatter graphs show how single points for each individual measurement's independent and dependent variables. Unlike a line graph, the points are not joined by lines. However, you may see a trend line or a line of best fit added to show the correlation between the two variables. And there are 5 types of correlation that you need to be able to recognize and describe as such. So we have positive correlation, both weak and strong, negative correlation, both weak and strong, and then we also have the situation of there being no correlation. So firstly, we have positive correlation. Positive correlation has a trend line that points up to the right. It doesn't matter how sharply it points up to the right. As long as it points up to the right, it is deemed positive correlation. And if there is a trend line drawn on the graph, you need to look for, does it point up or does it point downwards? Now, what positive correlation means is that as your independent variable is increasing, your dependent variable is also increasing. And we have 2 graphs here. One on the left, we've described that as a strong positive correlation, and the one on the right, we've described that as a weak positive correlation. Now, the strength of correlation is not related to how steep the line is. What determines whether or not a correlation is strong or weak are how close your points are to your trend line. If they are very close to the trend line, then you can say you have a strong positive correlation. If the points are spread out away from your trend line, then the correlation is weak. So positive correlation is just about the direction that your trend line is pointing. If it's pointing up to the right, it's positive. You judge the strength based on how close the points are to your trend line. If they are close, it is strong. If they are far away, it is weak. When it comes to negative correlation, we're looking for a trend line that points up to the left. And if that you have that trend line, what that indicates is that as your independent variable increases, your dependent variable decreases. And just like with positive correlation, it is not the steepness of the line that determines strength or weakness, it is how close your other points are to your trend line. If the points are close to the line, then it is a strong correlation. If they are far away from the line, then it is a weak correlation. The final situation is where we have no correlation. In a situation of no correlation, no trend line can be applied. And if you are looking at a graph with no trend line, with the data all spread out on it in your supplementary sheet, you could indicate that there is no correlation. What no correlation means is that the independent variable has no effect on the dependent variable. In all these graphs, I've used the word correlation. And if you're talking about correlation, what that means is you notice that as one thing changes, the other variable also changes. We cannot, just from one experiment on one graph, say anything more. We cannot say that because your independent variable changes, your dependent variable changes. So there is a distinct difference between correlation and causation. If there is a trend line, there is correlation. However, correlation just means you recognize a pattern between the two variables. You can only determine whether the independent variable has caused the change in the dependent variable if all other variables, including your confounding variables, have been controlled for within your experiment. And unless you have that data telling you that everything's been controlled for, you cannot talk about causation. So just be careful, and if you're given a graph, talk about that there is a correlation between the two variables. Do not say that one of them changes because the other one changes. And what I've got are a couple examples of different things that are correlated, but obviously not causing. So here's a graph showing from 1999 to 2009, two things. The US spending on science, space and technology, and the number of suicides by hanging, strangulation and suffocation in the US. And you can see that those two graphs are very highly correlated to one another. They almost have the same trend line. However, there is no way that because the US is spending more money on science, space and technology, that people are going to be hanging themselves more frequently. This is just a freak correlation rather than a causation. We see another one of these, looking at how much cheese was consumed in America between the years 2000 and 2009, and the number of people who died by becoming tangled in their bed sheets. Again, we can see quite a strong correlation between the two graphs. They're very, very close in trend line. However, there is no way that just because people eat more cheese that they have more chance of becoming tangled in their bed sheets. So be careful of using language that talks about one thing causing another thing to happen when all you have the evidence for is that they are correlated. When you see scatter plots and bar charts in Advanced Higher, you may also now see things called error bars. Error bars will stick up and down or left and right depending on the way that your graph is plotted. They represent the error in the measurement taken of your dependent variable. And they represent how confident you are and what range your measurement could actually have, anywhere from a lowest value to a highest value. The main thing you need to know about error bars is that if you have two bars on a bar chart or points on a scatter plot that have error bars and the error bars overlap, you can say that there is no significant difference between the results. That doesn't mean that the data cannot be used for anything, but it just means that if the error bars overlap, there is no significant difference between those results. So you cannot argue that 2 points are different if their error bars overlap. If we look at the bar chart on the left, many of those bars have error bar and many of those green bars have error bars on them that overlap with one another. However, if we look at the second bar and the third bar, so bar B and bar C, those are significantly different from one another because the error bars do not overlap. So you can make a conclusion about B versus C and see that B is definitely bigger than C. Again, B is significantly different than E because the error bars don't overlap, sorry. So you can say that B is significantly different than E. However, you cannot make a conclusion or say that C is significantly different than D or E because those error bars overlap with one another. The final type of graphical plot that you might get in Advanced Higher is a box and whisker plot. Box and whisker plots are a way of summarizing data, the whole set of data that you have. And they can either be drawn vertically or horizontally, as in the example shown there. Now, what these box and whisker plots have is they have a minimum and a maximum value. They have what is known as the lower quartile and the upper quartile, and in the middle they have something called the median. Now we'll go through what all these things mean. Now the max and min values tell you the largest and smaller data points collected during your experiment. We call the difference between these two numbers the range of the data. If the range is very high, then that could mean that your data is very reflective of the sample, or it could also indicate that there is a high degree of variation within your population. The minimum and maximum value are not necessarily used a lot for analysing data, or at least not for comparing one box and whisker plot to another. They look similar to error bars, but there are no error bars on a box and whisker plot. So if the maximum and minimum values overlap from one box and whisker plot to another, that does not mean you cannot. That does not mean that you can say that there is no significant difference between points. There is. There are no error bars on box and whisker plots. Next, we're going to look at the median and what is known as the interquartile range. Now, the median is the number that falls halfway between the largest and the smaller number. If you were to line up the numbers from the smallest to the largest and count halfway through them, the median is the number that falls in the middle there. The interquartile range is very similar to that. The lower quartile is if you take all the numbers, organize them from smallest to largest, and count 25% of the way across or a quarter of the way across. Whatever number is there is your lower quartile, and if you were to count 75% of the way, or 3/4 of the way through your data when it's organized from smallest to largest, that is what is your upper quartile. The difference between the lower quartile and the upper quartile is known as the interquartile range. If the interquartile range is big, it suggests that there is a large variation in your data. If it's small, it suggests that there is a small variation between your data points. If the median is exactly halfway between your interquartile range, it means your data is normally distributed and usually representative of a population. If your median value is closer to the lower quartile than the upper quartile, you describe that as the data being negatively skewed, which means within your data there are more smaller values or lower values and higher values, and the opposite is also true. If your median value is closer to the upper quartile, we describe the data as being positively skewed, meaning that you have more higher values or higher numbers than lower numbers. When it comes to median and mean, if your box plot is perfectly symmetrical, then you can make the argument that the mean and median values should be identical. If the median is skewed closer to the lower quartile, it means that the mean is higher than the median value. If your median is closer to the upper quartile than the middle, it means that your mean will be a lower value than your median. When it comes to analysing a single box plot, these are the list of things that the SQA have accepted as things to get 1 mark for an analysis. So the easiest thing is to say that the median value for this data is whatever number it is. You could also say, instead of quoting the median value, that more than 50% of samples have a value greater than the value of the median, or 50%. Or 75% of samples have a value greater than the lower quartile range, or 25% of samples have a value greater than the lower quartile range. Sorry, than the higher upper quartile range there. Or you can quote the range of the data from whatever the lowest value is to whatever the highest value is. There's quite a lot that you can give in terms of analysing a box plot. The final thing that you could be asked is, if you have multiple box and whisker plots, what would you be looking for when you're comparing data? There are 2 main things that they are looking for. 1, what are the values of the medians? If you've got 2 box and whisker plots, you can say that the median for this box and whisker plot is this value, and the median for this box and whisker plot is that value. Or you could make a comparative statement. The data on the left has a higher median than the data on the right. The other thing that they would look for you to compare is the size of the interquartile range. You work out the interquartile range by taking away the number for the lower quartile for the number for the upper quartile, and then you can compare the size of those 2 things to one another. So that has been the different types of data, the way that it can be represented and drawn, and how to interpret and analyse data in different forms.